I'm Vignesh Nidarshan. Today I'm going to explain about expression of CotA Laccase in E. coli for bioremediations. In here, aim of the project is to express the CotA Laccase enzyme from the Bacillus subtiles in Escherichia coli to produce an efficient biocatalyst for bioremediation application. Laccases are multi-copper oxidases that catalyze the oxidation of various organic and inorganic pollutions, including including phenolic compounds, synthetic dyes, and pharmaceuticals by optimizing E. coli as a heterologous host. This study aims to enhance the protection and activity of CotA A for potential use in environmental cleanup. These enzymes involved in the oxidation of phenolic and aromatic compound, which has the application in biodegradation of the toxic organic pollution such as dyes, pesticides, and pharmaceutical waste. This is the example of the CotA gene sequence cDNA from the Bacillus subtilis. In here, I have chosen PET28AA plus expression plasmid dye to the reasons such as strong TT promoter, Kistek for purification, multi-cloning site, and kanamycin resistance. These are the process of expression of plasmid. First one is the PCR amplification of CotA gene. Second process is the digestion with restriction enzymes. In here, both PCR product and PET28A plus plasmid with the chosen enzymes. The next step process is ligation into PET28A plus, then transformation into E. coli DH5M alba, then colony screening. In here, use colony PCR and restriction dietitian to confirm correct initiation. The final process is expression in E. coli BL21 CTE3. These primers include flanking restriction enzyme sites, ribosomal binding site, forward primer NTL sites with RBS, other reverse primers exol site. In here, ligation structure PCR amplified CotA and PET28A plus are both digested with the NTL and exol restriction enzyme. The digested CotA fragment is ligated into the MCS of PET28A plus, ensuring correct orientation. Successfully, ligase ensures CotA is under the control of the TT promoter for high level expression in E. coli. This is the plasmid map for PET28A plus CotA, showing the key features such as TT promoter for high expression, his tech for easy purification, NTL, exalt restriction sites used for cloning, kanamycin restrictions genes for selections. Transformation and selection. First, prepare the competent E. coli cells. In here, keep cells on ice to maintain their competence. Then add ligation mixture and incubate on ice for 30 minutes to allow the DNA update. Then heat stroke transformation and plate on selective LB agar. In here, only colonies with PET28A plus will survive. Examining colonies. After the transformation, not all colonies will have the correct plasmid with the CotA successfully inserted. To confirm colony PCR and restriction test analysis and sequencing confirmation. After the selecting colonies using machine resistance, we need to confirm that the CotA gene was correctly inserted into the PET vector. This can be done through the following steps. The first one is the plasmid mini preparation. In here, pick the several transformed colonies and grow them in the 5 millimeter LB growth plus Hanamase. Then do the restrictions mapping to check if CotA was inserted correctly. Next, do the colony PCR juice CotA specifically primers to amplify the insert directly from colonies. Then Sanger sequencing for final confirmation. The final step is expression and functional verification in E. coli BL21. After confirming that the CotA gene is correctly inserted in 28 plus, the final step is transfer the vector into a bacterial expression strain and verify functional protein expression. These are the references I have used to collect the information, and thank you for everyone for watching my presentation.